Hey guys, all right, let's go through a skeletal muscle contraction. We're gonna start with a neuron, draw a nerve cell here, and this is the axon terminal of a long neuron. So we'll label this. And if a neuron gets a message from the brain that allows it to successfully reach positive 40 millivolts, then what happens is that calcium, which is sitting out here outside the neuron, this positive 40 millivolts opens up this little calcium door here so that the calcium comes in to the neuron. This is all calcium. And it's going to bind to a vesicle or a bunch of vesicles that are sitting there waiting full of acetylcholine. So remember that acetylcholine was made over here by an enzyme called CHAT. And CHAT uses a acetyl from a mitochondria and puts it together with a choline, which is recycled from old acetylcholine that gets pumped back in through the cholinergic reuptake pump on the neuron. So the acetyl and the choline are put together to make acetylcholine, and then it goes and it sits in this vesicle and waits until it gets a signal from the brain. The calcium binds to it, which mobilizes it to move to the cell membrane. So the vesicle moves to the cell membrane, and a membrane protein called SNARE helps the acetylcholine move through the plasma membrane and out into the synapse. So the synapse is this space here between a neuron and a muscle cell. So the acetylcholine comes out. Let me draw a muscle cell here. So this is our muscle cell. We'll label it here. This is a muscle cell, and the acetylcholine is going to bind to receptors and activate the muscle cell to contract. That's how it signals the muscle cell to contract. So this acetylcholine comes down and binds to an acetylcholine receptor. So this is a acetylcholine receptor that the acetylcholine is binding to. When acetylcholine binds to the receptor and the receptor is working properly, then it activates a signal which gets sent down a T tubule. And if it's successful, it gets to positive 40 millivolts, and that opens a door or a gate on the sarcoplasmic reticulum, which stores a bunch of calcium in here, right? So this calcium gate opens, so it opens this calcium gate, and calcium comes out. And the calcium is going to bind to troponin. So let's draw our myosin-actin complex here. So we draw the thick filament, which is myosin, and then we have our thin filament here, and there's a ton of myosin and actin in the cell. We're just drawing one little sarcomere here. And so here we have actin, and the myosin heads, when activated, bind to the actin and pull it together, but it doesn't want to do that all the time. So actin is covered up, those binding sites are covered up by another protein called tropomyosin. Tropomyosin. And tropomyosin has these other protein called troponin that sits on tropomyosin. It actually pins it down. But when calcium binds to troponin, so here's calcium, and when it binds to troponin, then the tropomyosin moves out of the way, and myosin heads are able to bind to the actin and pull the actin together. So the actin gets pulled together, not the myosin. That myosin stays where it is. The actin gets pulled together. So actin gets pulled together, which shortens the entire cell, which shortens all the fibers, which shortens the entire muscle. And when the muscle shortens, that is contraction. And hopefully you remember that this contraction requires ATP. And that's why you need energy and ATP when you work out. You also need ATP to relax the muscle. So there's a little calcium pump here. So this is the calcium pump. You could call it the calcium reuptake pump if you wanted to. I'm just going to call it a calcium pump, and it uses ATP to pump calcium back into the sarcoplasmic reticulum, and when that happens, you get relaxation. So just to review, we have contraction. Contraction requires ATP, and that is when, well, I guess we could write down the whole story here. Let's do that over here. So the brain sends a signal to a nerve, which in this case reaches successfully positive 40 millivolts. So calcium enters the axon terminal, or the end of the neuron. The end of the neuron, causing acetylcholine to move to the cell membrane. And just to remember that the acetylcholine was made by chat in the neuron. So we have the acetylcholine sitting on the cell membrane and another protein called SNARE. So the SNARE protein helps acetylcholine leave the cell. So move across the cell membrane and leave the cell, which when something leaves the cell, we call it exocytosis, just like that. So acetylcholine is now out in the space between them, enters the synapse. Then acetylcholine binds to the acetylcholine receptor, but does not ever enter the muscle cell, right? So what is the fate of acetylcholine then? if it doesn't enter the muscle cell, well, we have a enzyme called acetylcholinesterase in it. I always draw her as Mrs. Pac-Man because what she goes along and she chews up the acetylcholine, chomp, 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 and turns the acetylcholine, gets broken up, and it gets, so the choline gets recycled back into the neuron, and the acetyl goes off and goes to a mitochondria. It could go to this mitochondria, it could go to mitochondria here, it could go to mitochondria anywhere, to be used for the Krebs cycle or to be used to make eventually another acetylcholine. The body is very good at recycling acetyl, right? So we could put that as a little side note that the acetylcholine is eaten or broken, the bond is broken by an enzyme called acetylcholine esterase. Okay, so that's acetylcholinesterase right there. Mrs. Pac-Man. And let's see. So let's go back to what's happening with our acetylcholine that bound to the receptor. So, the acetylcholine receptor activates the T tubule, right? And the T tubule reaches positive 40 millivolts to open the calcium door on the sarcoplasmic reticulum. So then the calcium, this is a 7, this is an 8, so the calcium diffuses out of the sarcoplasmic reticulum, and it goes into the cytoplasm of the muscle cell. I'll just say cell because I'm running out of room. And that calcium binds to troponin, which is a protein, which causes tropomyosin to move out of the way, exposing the binding sites for the myosin heads. And the myosin heads bind to actin, and here neatly on number 12, it pulls actin together with, it's very active, so it's with ATP, right? So overall, over here, in the contraction, is that the cell, the calcium is going to be in the cytoplasm, and relaxation, the calcium will be in the cytoplasm. I'm sorry, in the sarcoplasmic reticulum. Sarcoplasmic reticulum. Oh my gosh. There we go. Really having trouble spelling that. All right, now there's a couple things that can go wrong in this, okay? First of all, maybe the acetylcholine receptors aren't working properly, and that is a disease called Let's see if I can write it up here if you can see it. So this is when it goes wrong, right? So these are some pathophysiology, some disorders. Okay, so we have myasthenia gravis, which some of you may have heard before. And this is a autoimmune disease. Autoimmune disease where the patient's own antibodies, which are made by their immune cells, attack the acetylcholine receptors on their muscle cells, okay? And this is going to cause, as you can imagine, think about it. If acetylcholine is made, but the receptor's not working, the muscle's not going to contract, and you might get some contraction, but not a lot. So it's going to cause muscle weakness, and fatigue, muscle fatigue. Muscles get tired easier. And interestingly, as some of you might know, this starts in the muscles of the face. They're not really sure why, but the person's face will begin to droop, especially their mouth and their eyes. They'll begin to get weak and droop. And so if we wanted to draw what this looked like, so this would be myasthenia gravis we're drawing here. So we have an acetylcholine receptor, and a antibody looks like a letter Y, kind of like this. So this is an antibody. So this is an antibody, and it is attacking an acetylcholine receptor. So it's going to shoot little chemical bullets, pew, pew, and kill it. Sorry, it's very late on a Sunday. But these bullets, these are true things. They're little perforins, and they actually puncture holes in the plasma, the phospholipid bilayer, so that it's not functioning, and that is myasthenia gravis. There is another disease, which is called muscular dystrophy. Let's see, let's do that one down here where we have a little room. So we have muscular dystrophy, and I guarantee that you have all seen someone with muscular dystrophy. Muscular dystrophy is a inherited disease. It is a mutated copy of the gene for a protein called dystrophin, dystrophin. So dystrophin, right? And this dystrophin protein normally plugs the cell membrane, so calcium can't leak. It also attaches to the cytoskeleton to provide strength and structure and function. And it kind of sits in there like this. So it sits here and it attaches to the cytoskeleton like that. And this is dystrophin. So if that's messed up with muscular dystrophy, then the muscles, of course, begin to not be as strong. They kind of fold in on themselves. So the muscles break down. And I encourage you to look up pictures of muscular dystrophy. I encourage you to look up pictures of myasthenia gravis and do some research. If you look into this and just read a little bit about it, you will understand it more, and you will ultimately understand all of this better, and you'll be able to make those connections better. Another thing is Botox. We talked about Botox and how Botox cuts the snare protein so that the acetylcholine is not released. So acetylcholine is not released. So no contraction. Interesting that Botox stops it here. Myasthenia gravis stops it here at the acetylcholine receptor. Muscular dystrophy is just, it's just going to be an overall breakdown of the muscle fiber. Completely breaks down. So let's talk about what type of medicine. Let's see. I have a little space right here. What type of medicine is given to people when we want to increase the amount of acetylcholine here? What we would do is give them a medicine that's going to inhibit or stop this acetylcholinesterase. And this medicine is called, so this is a medicine, a medicine called acetylcholinesterase inhibitor. And what that's going to do is block the action of acetylcholinesterase. Mrs. Pac-Man. So think about it. If she is not breaking down acetylcholine as much, then you're going to have acetylcholine build up. So acetylcholine will increase in the synapse. So why in the world would we want to increase acetylcholine here? Wouldn't that cause it to constantly stimulate the muscle cell and cause contraction? And yes, unfortunately, that is a side effect of this medicine. But what these patients have is so much worse than that they need it, because these are for patients. These are patients with Alzheimer's. Oh my gosh, spelling Alzheimer's wrong. Alzheimer's disease. And so they're trying to increase stimulation between the neurons in the brain, but as a side effect, it does cause overstimulation of muscle cells. And so as a side effect, they get twitching when they're taking this medicine. And I'm going to stop here and go over the cardiac muscle cell. But a good way to study this would be to look over your doodle notes. Make sure that you know all the words on those doodle notes and are familiar with how they apply to the physiology of skeletal muscle contraction.